Okay, so in this short EdPuzzle lecture, we're going to be looking at the structure of carbon and how and why it contributes to the formation of carbon-based life as we know it, or organic life as we know it. How do you get large complex molecules that are carbon-based? We're going to talk about what a functional group is and how they affect the diversity and function of organic molecules. Basically, how do they help determine what bonds form and with which kinds of compounds and what kinds of bonds form. And of course, as we know, that's going to affect the shape of molecules. And we know the shape and the function of molecules are directly related to one another. We will also be covering the important functional groups that you will be seeing repeatedly in lots of different molecules for the rest of the course. So now is a good time to learn those and to learn their properties, and then you can recognize them easily later on and understand exactly how they're going to work when they come in contact with other molecules. Okay, so why is carbon so important? Why are we talking so much about carbon? Carbon is actually not terribly plentiful on Earth. It is the fourth most common element in the universe, but we don't have quite as much carbon. It's about the 14th or 15th most plentiful element on the planet. So the question, of course, becomes, why is carbon so important for life as we know it? And a big part of that reason is carbon's ability to form stable bonds, both with itself and other atoms. And in fact, most of our organic compounds or molecules, and organic just means, for our purposes anyway, it just means that it contains carbon. So we're using the chemical definition of organic, right? So when we're talking about organic chemistry, we're talking about chemistry that involves carbon. This has nothing to do, right, with organic, with the word authentic or with organic farming or anything like that, which means, of course, no pesticides or chemical additives. So totally different meaning to the same word, which of course happens in biology quite often and is confusing. So when we're talking about organic, for our purposes, we're talking about just means, hey, it contains carbon, which is almost every molecule that we're really going to talk about in this course, besides water, in terms of its biological importance. And so, again, you see carbon bonded to itself in long chains and ring structures or other forms of molecules. It also very often is bonded to oxygen, hydrogen, nitrogen, and so on to form very large, complex molecules. Okay, if I can get my slide to advance, maybe not. So what is this? I can draw or I can... What if I don't want to use any of my pointy tools? There, Jiminy Christmas. I'll have to edit that part. Okay. So the reason that carbon can form these complex bonds with other atoms, of course, has to do with how many electrons it has in the valence shell, right? Remembering from last week, that is the outermost shell of electrons that are circling the core of the atom. In the case of hydrogen, right, hydrogen has one proton in its nucleus. and it's got one electron. So it is balanced in terms of charge, but its outer valence, as you can see on the dot structure there at the bottom of your screen, right down here at the bottom, right? So our Lewis dot structure here, it has one dot. That tells you that it has one electron. Well, right, remember our valence wants to be balanced, right? So electrons spin in the opposite direction. So one spins to the left and the other to the right, or up or down. And so to be balanced, they really want to have two. And so this is why hydrogen, even though it is neutral in terms of charge, it likes to form bonds and very stable covalent bonds with itself in nature, right? You generally just see H alone, you see H2, right? If you see a hydrogen by itself, typically it's in the form of H plus. It's an ion, meaning that it has lost the electron, right? So it's positively charged. And it's basically just a proton floating around. But it's kind of happy in that form because, I mean, obviously it wants to be, it wants to neutralize the charge, but there's no electrons in its outer valence. So it's not unbalanced, as it were. Likewise, oxygen, as we can see again from the dot structure, has 6 electrons, but it has basically 2 bonding sites. Because it really wants to have 8, right? And we know oxygen is extremely electronegative, and so it's very greedy, and so it will form bonds very easily with other atoms, and then of course hogs the electrons, as we know, right? So this is why we see H2O, for example, right? Because you form a bond with 2 hydrogens, and that way you get a neutral molecule. It's polar, but it's not an ion. It doesn't have a charge on it, and all of the atoms involved have electrons to fill their outer valence shell, and they're happy, right? And so carbon is important because of the number and type of bonds that it can form with both itself and different atoms. Okay, so first we're going to look at the kinds of bonds that carbon forms with itself. So again, it can bond to itself in a single bond. It can bond with itself repeatedly in a single bond to form a long chain. You can get a branched chain depending on where, basically how it bonds with itself. So something like isobutane. Over here in the middle, where a carbon has bonded to itself as single bond. Now you'll notice there's a few other places where carbon is double bonded to itself, both forms of butene. On the top right there, for example, involve a double bond, right? And so we know every time you see a double bond, right, that tells you that those 2 atoms are sharing 2 pairs of electrons. And of course, carbon can form 4 bonds total because it has 4 valence electrons. So it can, we can see with ethane, each carbon is bonded to the other carbon, and then to 3 hydrogens for a total of 4 bonds for each of those carbon atoms. Anytime you see a carbon double bonded to something else, that means that sometimes it's carbon, sometimes it's oxygen, sometimes it's something else. Again, that means they're sharing 2 pairs of electrons. And it also means that you have the opportunity for differences in the structure of the molecule in terms of where the other atoms are that are bonded to the carbon. So for example, in 2-butene, the H's are all on you have H's on basically the top of both, but then you notice on the bottom, right, depending on where the double bond is, you can either have your hydrogens at the ends of the molecule or on one side or on the other side. if you're, 1-butene versus 2-butene. And that just tells you where the double bond is located. And we're going to talk about the how the position of a double bond affects the structure of a molecule, the 3D spatial structure of the molecule here in just a second. And I also want to take a moment to point out the ring structure. So we have we can form chains if we're carbon, but we can also form ring structures. And again, that can involve a single bond, like in cyclohexane, or a double bond between the carbon atoms, as in benzene. And of course, we're going to see that ring structure coming up, and it's very, very important to the structure of glucose. It's very important to the structure of our DNA and RNA bases that we're going to be looking at here in just a minute, both in terms of the sugar structure and also the nitrogenous bases that are involved in the structure of DNA. So the fact that basically carbon is super flexible in terms of what kinds of atoms it can bond to and what kinds of bonds it can form both with other atoms and itself, with a maximum of 4. Okay. And these molecules, of course, all here are just involving carbon and hydrogen. So quite often we name molecules based on their component pieces. What exactly are they made up of, right? If it's made of phosphorus, right, for example, we call it a phosphate. If it involves sulfur, we call it a sulfate, and so on. Okay, so here we can look at how the 3D structure of the molecule is affected by the number and position of the bonds between the carbon molecules. Excuse me, between the carbon atoms. I keep saying molecules. Sorry about that. So a super simple organic molecule like methane, right? You have all 4 bonding sites are full, but they're full of the same atom. And so when you can see the ball and stick model, you get something that kind of looks like a pyramid shaped molecule in terms of how that looks in 3D space. Ethane, for example, though, you get basically 2 pyramids that are kind of stuck together. You get this kind of odd-shaped molecule. But what's interesting, anytime you see a single bond, right, the molecules can actually spin in 3D space because they're only held together by 1 pair of electrons. And so they can kind of rotate independently of each other. When you look at ethene here at the bottom, you have a double bond between the 2 carbons there. And you can see on the ball and stick model how that locks the molecule into one plane, right? You can twist it a little bit. I mean, imagine if those 2 the double bond there in the middle was sort of like little pieces of rubber or something like that. You can twist it a little bit, but you can't spin them independently. You can't rotate them around and keep the other side of the molecule stationary, right? Because they are, because you have 2 pairs of electrons involved, it locks the whole molecule into one plane. And that's going to become important later on when we look at, again, the structure and function of molecules. So where any double bonds are located, whether they're present or not, and of course what atoms are involved, all those things are going to affect the structure of a molecule. And we know, right, that the structure of a molecule and its job are directly related to each other, right? What kinds of molecules it can react with is going to be really determined by the structure of that particular molecule, and that's going to become really super important when we talk about enzymes, because enzymes bind with very specific compounds or molecules, and in a fashion that's almost like a lock and a key. It's really specific what they will bind to in particular forms. And so essentially, if you bind something else to a molecule, it's going to change the shape of the molecule, and that molecule may or may not be able to, probably not, be able to do the job that it was originally, supposed to do, based on its structure. So it's something we talked about earlier when we were looking at uh, just general introduction to biological chemistry. Okay, so because carbon can form bonds with 4 other atoms, right? And those atoms might be pretty much anything. Most commonly, again, they're hydrogen, oxygen, nitrogen, and itself. But because of this, we can actually get compounds that have the same molecular formula, meaning the same number and type of atoms involved, but they have different structures. And therefore, because they have different structures, they have different properties. And we call these isomers. And we have several different kinds of isomers that are possible. And again, for folks who are taking chemistry or have taken chemistry, you're probably going to be pretty familiar with this. A structural isomer is, of course, two molecules that have the same molecular formula, but their bonds are different or in the different place. Right? So, for example, 2 examples of the structural isomers are pentane and 2-methylbutane. So they have the same molecular formula, the same number of carbons, the same number of hydrogens. Those are the only 2 atoms involved, but one is a chain, and the other is kind of a T-shaped molecule. So that's an example of a structural isomer. You can also get geometric isomers. And again, these are molecules that have the same number of atoms, the same atoms involved, right? The same types of atoms involved, the same molecular formula. And in the case of a geometric isomer, the bond is in the same place. However, how the atoms are, which atoms are bonded to which, are different. So here we've got a cis isomer on the left-hand side, and you'll notice that the hydrogens are on the same side of the molecule. They're on one side, and the X's, which could be single atoms, or it could be another small molecule, something that's bonded to that carbon, but they're both on the same side of the molecule. And you're probably going, well, how do I know which side it is, right? Remember, if you've got a double bond there between those carbons, that locks them into the same plane, right? So they can't twist and rotate. You can't just spin that right-hand side carbon so that the X is on the bottom and the H is on the top. That's not possible with the way that the atoms are bonded. So you're locked in place. And this, of course, means that you could have 2 atoms or molecules that are on the same side that are the same, or different, but you could have them basically, if they're both on the same side, that's a cis isomer. And again, they have to be molecules that are the same molecular formula, right? So you can't just go, oh, they're sort of the same. No, they have to have the exact same molecular formula. In the case of a geometric isomer, they also have the same bond placement, but where the other atoms are bonded is different. So the trans isomer on the right-hand side, that just means they're on the opposite sides, right? So they're across the molecule from each other instead of being on the same side. And so you can imagine if that X is maybe an oxygen or an OH, or even a carbon bonded to a few other hydrogens, that's going to give the different properties to different isomers depending on whether those groups are on the same side as each other. They may interact with each other, especially if they're polar, or or different properties based on if they're on the opposite side. And that's going to become really important when we look at our polymers. Later on, we're going to look at what happens when we string a bunch of the same molecules together to form very long compounds. If all of the groups are on the same side as the double bond, they're going to interact with each other in particular ways, and it's going to be different if they're on the opposite side. So geometric isomers can be really, really important. And then the last kind of isomer that we'll talk about are enantiomers. And an enantiomer, again, are two molecules that have the same molecular formula, but in this case, the molecules are mirror images of each other. And that's possible only because, again, carbon can bond to 4 other atoms. And it's only possible to have an enantiomer where you have 2 isomers that have the same molecular formula if there are 4 different atoms or molecules bonded to that carbon, right? So here we have the isomer on the left, right? These guys you have 4 different things that are bonded, and because they're all different, then you can form a mirror image of each other. So again, remember, you can't necessarily spin things around. Oh, I'll just flip it around upside down. No, that's not how it works. Remember, this is a mirror image, just like our hands. You can line them up, like you're clapping, but really a mirror image is going to be the same but the opposite, right here. So right, for example, an L isomer, you'll notice the thumb's on the left. In the D isomer, the thumb is on the right. And so that's essentially what you're generating when you have an asymmetric carbon, a carbon that's bonded to 4 different atoms, 4 different things, right? Different atoms or molecules. You can end up with a mirror image. And those have very specific, or can have very specific properties and very different properties. Again, because they're mirror images, they're not going to bind with other molecules in the same way. The isomers are going to act differently from each other. So because the structure of enantiomers is not exactly the same, right? They're not identical, they're mirror images of each other, right? So they have the same molecular formula, but different placements of the atoms relative to each other. So you essentially, like I said, have a mirror image, and that means that they are not going to bind to other molecules in the same way as each other. So for example, ibuprofen that you get in your bottle of Advil or whatever you buy at the corner pharmacy is going to be a mix of the enantiomers. So you have the effective version. This is the one that actually binds to pain receptors and actually is the one that's causing or enacting the pain relief by binding with different receptors or molecules. Then you have the ineffective enantiomer. In this case, our ibuprofen, I don't want to say does nothing, but it pretty much does nothing for pain. And now it can reverse, basically it can spontaneously convert from one enantiomer to the other as molecules move around and bonds change. So it actually will spontaneously convert from the R isomer to the S isomer. But even the R isomer has been shown to slightly enhance the effectiveness of the S isomer by how it binds with other kinds of enzymes in the body. But it doesn't really do anything directly for pain relief. So, but because it doesn't have any negative effects, your pharmaceutical company, whoever's manufacturing this, will just let the two mix together and they don't really care, right? Now, albuterol is a different story. So this is, it exists again in 2 forms, 2 isomers, 2 enantiomers. You have R-albuterol and S-albuterol. R-albuterol, of course, is the effective form. Anybody who has asthma or knows someone who has asthma may know this is the effective ingredient in a lot of inhalers, specifically rescue inhalers. And albuterol essentially it binds to receptors in your bronchial tubes and causes them to essentially expand. So essentially opening up those airways. Now that's what R-albuterol does. S-albuterol essentially neutralizes R-albuterol. So essentially it cancels it out. And so, of course, you can imagine what you don't want to put in someone's rescue inhaler, right, is a mix of R and S albuterol. You don't want the two enantiomers in there together because one's going to cancel the other out, and that inhaler is going to do absolutely nothing. So what they have to do when they are manufacturing albuterol for these kinds of rescue inhalers and for other applications, of course, is you have to do additional chemical reactions to distill out or remove the ineffective enantiomer form. So you have to get rid of the S-albuterol before you package that and send it off and send it out somewhere as an effective medication. So you don't want them together. And that's actually true for a lot of different kinds of enantiomers. You may have an effective form that does a particular job, and the other one either sometimes has effectiveness or sometimes it doesn't do anything because, again, they're mirror images. They're not exactly the same. Or in some cases, that it has quite harmful side effects. So you need to be cognizant of that. Okay, so the structure of our organic molecules, and again, remembering organic just means it's got carbon, right? So it depends on the properties of organic molecules. Basically, what they do, how they interact with other molecules, depend on the structure of the carbon skeleton, which is what we've just been talking about, right? Different kinds of isomers and so on. That all has to do with how atoms are bonded to the carbon, where the bonds are between the carbon molecules, and how the placement of the carbon molecules, the structure of that carbon skeleton. And the properties of organic molecules also depend on the properties of the other molecules that are bonded to that carbon, right? So it depends on the chemical components that are involved in those reactions. And those are usually the other molecules that are bonded to the carbon. And many of those fall into what we call functional groups. And functional groups have particular properties, which is why we sort of lump them into functional groups. And then based on those common properties, we can predict exactly how a molecule will behave. For example, when we put it in aqueous solution, or when we introduce it to other types of molecules with particular properties, we can predict how those things are going to react. And that helps us, again, to determine the function of a particular molecule. So I know there's 7 of these. We're going to go through them one at a time. Okay, so generally we're going to start with our functional groups, and we're going to go from simpler to more complex. So one of our simplest functional groups is basically an oxygen bonded to a hydrogen. And those two things are then bonded to a carbon. The OH group is also known as the alcohol group. So most of the compounds involving them end their name in OL, right? So isopropyl. Trying to think of another one. Ethanol, right? Here we go, right in front of me. Isopropyl alcohol. Ethanol, methanol with an M, right? All those kinds of things are alcohols. And that just means that they have hydroxyl groups. So you have a single oxygen atom, again, bonded to a carbon and a hydrogen. So it's the OH group is the functional group involved there. Okay, so another kind of functional group that you're likely to encounter is the carbonyl. And this is basically a carbon that's double bonded to just an oxygen. And the difference, of course, as we're seeing between a ketone and an aldehyde is essentially where that carbonyl occurs within a particular molecule. So if it happens at the end, like we see with propanol here, where you're the carbon double bonded to the oxygen, so there's your carbonyl, which is in that little dark blue box. And then the carbon is then also bonded to another carbon as well as a hydrogen. In acetone, the carbonyl is in the middle of the molecule, and so you can see as a consequence of that double bond there that causes the rest of the molecule to kind of be bent. It changes the shape of the molecule, and that means it's going to change how that molecule interacts with other molecules. So in aldehydes, the carbonyl is at the end, and in ketones, the carbonyl is somewhere in the middle of the overall carbon skeleton. Now, of course, you're seeing the oxygen, so you're probably thinking, okay, I know oxygen's very electronegative, and because it's sharing electrons with the carbon, that sharing is likely to be unequal. Which means you are likely to have a slightly polar molecule, which of course means that carbonyls usually play nicely with water, right? So they're generally going to dissolve and be just fine in an aqueous solution. Also interesting, here acetone and propanol, you probably have noticed, have the same molecular formula. But obviously the structure of the molecule is different because of where that carbonyl is located. Okay, another functional group that we are going to be spending a lot of time with these guys. They come up all the time, is a carboxyl. And what you've probably already noticed as well, I'm seeing a hydroxyl, right? That's the OH, that's the alcohol group. And then I'm also seeing a carbonyl group. And so basically a carboxyl, as you know, it's just like I took part of one and part of the other and I smushed them together, right? So yeah, I got the carbo from the carbonyl and I've got the oxyl from hydroxyl and I stuck them together. So a carboxyl is a carbon double bonded to an oxygen, and then that carbon is also bonded to a hydroxyl group. We call our carboxyls quite often acid groups because, as we're going to see when we talk about acids and bases, an acid in aqueous solution, an acid molecule donates a hydrogen ion or a proton, same thing. That's the H plus there. It donates that to the solution. And by doing so, it causes the pH to go down as that solution becomes more acidic. So you'll also notice, let me get my little pointer doodad here, right? So here's my H plus, right? And this is going to basically make it more acidic. That's changing the pH by its presence. And then you'll also notice, right, we've got a two-way arrow here, right? So it's going back and forth. And so what that means, right, is that acetic acid, when placed in aqueous solution, is in a dynamic equilibrium between the acetic acid form and the acetate ion. So basically that means that H plus and the acetate are spontaneously recombining to form acetic acid, and then acetic acid is spontaneously dissociating or breaking apart to form acetate and the H plus. And so it's a chemical reaction that can go back and forth depending on the conditions. But in general, when you put an acid group in aqueous solution, it dissociates. It donates a proton or an H plus ion to the solution, which of course affects the pH. And of course, oxygen being greedy, right? You'll notice it kept the electron, right? So that oxygen there is then negatively charged. So carboxyls, of course, play very, very nicely with water because of that. So it's very, very polar interaction here with the OH in the hydroxyl group. So much so that in aqueous solution, again, it breaks apart. Trying to think if there's anything else I want to say about carboxyls. I can't think of anything off the top of my head. Okay, another important functional group is the amino group, or amines. And amines or aminos, right, are nitrogen based group. So we have our NH2 here. So our amino group is attached in this case to a carbon and some other stuff. What to know about amino groups have basically the opposite behavior of a carboxyl, right? A carboxyl is an acid group. Acids donate protons to the solution. Well, amino groups act like a base, and they accept or pick up a H plus ion from the surrounding solution, which is usually water. And that means that, again, they pick up an H plus, so they picked up this positive charge and this extra proton to form NH3 from NH2, so NH3 plus. So again, it's going to be charged in aqueous solution, and it means it's going to, again, it's acted like a base, and that means it's also going to play very nicely with water, and it's also going to be attracted to anything that is negatively charged, right? And this is going to affect how amino groups bond with other molecules. Something you're going to notice here, right? Our example of glycine, right? So I have an amino group, right? I've got my carbon with its 4 bonds, and the carbon on the other side is bonded to, right, this thing, which is a carboxyl group. So I have an acid group, and I have an amino group, which means I have an amino acid. And glycine is an amino acid. It's one of the simplest amino acids because the carbon is bounded to the amino group, the acid group, and then the others are just hydrogens, just plain old hydrogens. Other amino acids will have what's called the R group here at the top, and it will be something else. And sometimes they can be quite complicated, sometimes they're very simple. This is sort of one of the simplest ones. Okay, so another functional group that we really need to cover, mostly because of its role in the formation of protein structure, is the sulfhydryl group. And this is pretty simple. It's just a sulfur bonded to a hydrogen. It may also be written as HS, depending on how it goes. And of course, you can see it here in cysteine, right? So here's, again, another amino acid, right? Here's our amino group, and here's our carboxyl group, right? And then here is the sulfhydryl group is right there. Whoa! Got a little carried away there. Okay. So anyway, there's our sulfhydryl group is right there. Again, the reason I bring this up is these are bonds that can form between sulfhydryl functional groups are called disulfide bonds or disulfide linkages, and they're a very strong covalent bond. And so they really have an important role in protein structure for a lot of different kinds of protein-based molecules. As a fun fact, right, this is cross-linking cysteine, which is an amino acid, right? So which is a building block of protein. So it's found in our hair, on our head. And so what we can do chemically is break these disulfide bonds, straighten our hair, or curl it around something, right? Piece of metal, piece of plastic, something, and then apply another chemical to essentially reinstate those disulfide bonds. And this, of course, is the process of giving someone a perm, right? Or a permanent wave, if anyone's had a perm. Or, of course, if you watched Legally Blonde, which was on Netflix not too long ago. Right? It's the cardinal rule for perm maintenance. You don't wet your hair within, I think it's 24 hours, but it might be 72, at the risk of deactivating the ammonium thioglycolate, right? So that's of course the chemical that is reinstating the disulfide bridges between the amino acids in your hair and allowing it to stay always curly, always in that particular shape. But it takes a while to set everything. So essentially get those bonds to reform. And then once they reform, right, you leave them alone and then you've got curly hair, right? So fun fact. Okay, second to last functional group, I swear, I promise we're almost done. So is the phosphate. This is an extraordinarily important functional group. And as you'll notice, we have a phosphorus atom surrounded by oxygen. Yikes, all the way around, right? And even the oxygens here that are only single bonded to the phosphorus have a negative charge on them. So you can imagine if you're trying to bond something else to a phosphate, like another phosphate, those negative charges are going to repulse each other. They don't want to be next to each other, right? And again, you have an enzyme that helps facilitate this process, and you can put two of them together. Putting a third one together takes a pretty good amount of energy, again, on the cellular level. It can be done, but it's very unstable. And the reason I bring this up, right, is having three phosphate groups linked together is part of a molecule called adenosine triphosphate, or we just abbreviate that ATP. And this is our essentially energy cache molecule that our cells use all the time to essentially perform work, right? And so we're going to be spending a lot of time talking about ATP and the energetics of cellular metabolism here pretty soon. But just a reminder, right? The ATP is made up of adenosine, which is essentially a nucleic acid base, not an acid base as an acidic, but it's nucleic acid because it has an acid group on it. And we call them bases. They're fundamental components of DNA. So it's one of those linked to three phosphate groups. So again, fairly unstable. It wants to break apart, and when it does break apart, it releases a lot of energy, and that's the energy that can be used to move things around. And it reacts with water very, very easily within our cells. So you can imagine, again, all this oxygen, you're probably thinking, okay, that's going to play with water really easily. It likes to be with water, and you're absolutely right, because you see that phosphate groups are also present on one end of phospholipids, right? So you have a phosphate group on one end that likes to play with water, and then you have a hydrocarbon tail on the other end that really does not like to play with water. But if you put phospholipids in aqueous solution, those hydrocarbon tails want to get together, and the phosphate groups want to face away into the aqueous solution. And so what spontaneously forms is a lipid bilayer, which at its very basic level is essentially the components of our cell membranes. So really basic building blocks there. Ooh, all right, we made it, last one. Okay, and that is a very simple functional group, and that's the methyl group. And you can notice its formula is very, very similar to methane, right, which is CH4, and it's just carbon and hydrogen. no oxygen to be found, right? No phosphorus, no sulfur, no nitrogen, just carbon and hydrogen. So this is very much a non-polar molecule. And where you tend to see these are, again, have non-polar behavior. So they like to associate with other non-polar molecules like hydrocarbons and lipids and things like that. And they don't like to associate with anything polar or charged, water, any of that stuff. So we're going to be looking at the role of methyl groups. They also have an important role in the regulation of gene expression that we're going to be looking at later on. Our cells use methyl groups to help essentially permanently turn genes off by attaching them to certain areas of our genome. But for now, we'll mainly be focusing on its non-water loving, right? So it's water hating properties.